Hello, guys. Perfect. I think I'll just give everyone just 1 or 2 more minutes before we start this webinar. Glad to see so many different kind of peoples here. I'll also make sure that we just double check that everyone has the access to the meeting also. Right. I see that there will be a couple more people joining, but I can just start very, very briefly. My name is Daniel Kumar. I'm the CEO of ETN. ETN is the company that has commercialized the EME device, and it was founded in 2013 when Stig Pedersen Bjerregaard, the professor that invented EME, wanted to discuss how we could commercialize this, at that time, quite a novel invention, but that was still in the research phase. Last year, we're proud and lucky and glad to launch the actual instrument, the ETN12 EME. And today we will have an introduction to EME. This will last for around 30 minutes, and then we will have 15 minutes Q&A. If people want and have time, they can still be here a bit longer if they have more questions. Please just add the question in the Q&A or in the chat, and we will either just go through them together. Most likely, Frederick will kind of touch into some of these points during this presentation. Yeah, so with that, I think I will just hand the word over to Frederick.
Yeah, thank you, Daniel. I hope you can all see my screen. Short introduction to me. My name is Fredrik André Hansen, as it says here, and I am a postdoctoral researcher at the University of Oslo in the group called Sample Prep at UIO. And here we do research that is very focused on electromembrane extraction. So over the years, we have, of course, built up a big portfolio of knowledge and results. And for these first 30 minutes, I will give you an introduction to the EME principle and its advantages. We will cover the ETN instrument, consumables, and procedure, a bit about what kind of performance you get for EME, how you can get started, and then I have some few application examples. And if you have any questions, as Daniel mentioned, then please just post them in the Q&A section. So, electromembrane extraction, or EME for short, is a micro-extraction technique derived from liquid-liquid extraction. So this is a sample preparation procedure that essentially is a process of electrophoresis across an oil membrane. And this is in particularly directed towards application for HPLC and LC-MS analysis, but of course not limited to this. The principle itself was invented in 2006 here at University of Oslo, and to date more than 500 scientific papers have been published covering all kinds of different substances, matrices, and technical formats. In brief, EME is performed by taking a porous polymer membrane. This is the one we can see here, which essentially just is a piece of plastic with a lot of holes in it. This is quite thin. It's approximately 150 micrometers thick. And in this piece of plastic, we take a droplet of organic solvent and immobilize in the pores of this. And here it's the solvent sits with very strong capillary forces. so it's very stable in these pores. Then we use what is now called a supported liquid membrane, or SLM for short, to separate our sample. For example, it could be a biological sample from a clean acceptor solution that is just made of a neat buffer agent, for example, formic acid. Then we place these solutions inside conductive containers, and we make sure that the substance we're interested in has an ionization, positive or negative. Let's say we are interested in extracting the positive ion. Then we apply a negative voltage to the acceptor solution and a positive to the sample solution. And in this way, we can stimulate the ion to migrate towards the negative electrode across this supported liquid membrane. And as mentioned, this has been performed in over 500 scientific papers, but now is the first time you can actually buy equipment to perform this, which of course brings the standard of methods to a whole new level. The advantages of EME include, and I would say this is, in my opinion, the main advantage, is that it is a one-step sample preparation technique, meaning that you can load your sample, let's say it could be whole blood, into the sample vial, and then you do the extraction, and on the other side you have the acceptor solution, which is aqueous and can go directly into the LC-MS instrument without having to do any evaporation, reconstitution steps that can be time consuming. In addition, the sample does not have to be pre-treated in any way, so you don't have to do filtrations or centrifugations or other things that might be in a normal workflow. Further, the technique is compatible with quite complex samples. I mentioned whole blood, and then of course serum or plasma, urine, cerebrospinal fluid, or even tissue is possible, as you will see towards the end. Despite being compatible with very complex matrices, then you can get very pure extracts. And this is because of the selectivity is derived from both the analyte charge. So you can see here that in this case, the positive ions are extracted, while the negative ions remain because of the direction of the electric field, and neutral substances are not affected by the electric field, so they just distribute to some extent between the sample and this small organic phase that is the supported liquid membrane. And this means that we can not only separate ions based on charge, but also the nature of the hydrophobic solvent discriminates based on hydrophobicity, which you will also see. So this means that we get quite pure extracts. And this involves the complete discrimination of proteins and phospholipids that, when you consider, for example, a blood-based matrix, are often the most troublesome components for LC-MS. The technique is further non-destructive in the sense that once you have extracted the sample, you can reuse it for something else. And we use green and safe chemistry. Green in the sense that we use very little organic solvent, and safe by the solvent that we do use is non-volatile and non-toxic. So you can work with this out in the open. You don't have to work in a fume hood. So just to summarize, when is it in particular you would use electromembrane extraction? Well, since we rely on an electric fields, then you need ionizable analytes. You can also use it when you want a simple one-step workflow and when you want or need excellent cleanup and selectivity. And in the past, what people have used it for is captured in this figure, where you can see the different matrices, so a lot of biological fluids, but also wastewater, reaction mixtures, or food has been analyzed. And in particular, it's been used for pharmaceutical drugs, endogenous metabolites, heavy metals, peptides, and some preparative applications. Then we come to the instrument itself and the consumables. Here you see the base consumables, which is conductive vials. These are the black ones you see here. These are in the shape of a standard HPLC vial. but you can see that there is a black part and a white part. And the black part is polypropylene with some conductive material added, and this gives us the ability to conduct electricity. And the transparent or white part here is then just to make the vial standard size. You have a lead-tight interface where the vials are screwed into, and in this interface you also have a polypropylene membrane which is used to house this thin supported liquid membrane that is the organic solvents. The vials come in two different sizes, one that can hold up to 200 microliters and one that can hold up to 600. But because it's most efficient not to fill them all the way up, the working volume is between 200 and 500 microliter for the large one and 30 to 150 for the small one. So you can imagine that if you take a large vial for your sample and a small one for the acceptor, then you can get up to about 10-fold pre-concentration, which is the limit in the current formats. When you have assembled one unit, then it looks like this, where you have the sample on one side and the acceptor on the other side, and then this supported liquid membrane in between. Then you can take this unit and what else samples you have and place inside the holder of the instrument, where you can see there is space for 12 positions here. Then you cover these with a lid. That is the one up here, and this lid has space for, not space, it has one printed electrode that will come into contact with each vial, so you can close the electric circuits. The whole instrument is seen here, which has a quite small footprint, so it doesn't take up a lot of space in the lab. The parameters of the instrument and control is then controlled by a software that is controlled using a PC connected to. We will not go too much into details with this software. That might be the subject of a future webinar. So how do you do it practically? The first thing you do is that you have to get the polypropylene membrane into the union. These come in cards of 10 membranes. So you pull one out and then you place it on the top of the union. And then it's often easy just to use a pipette tip to press it into place. And then it will sit down in the middle, as you can see here. Then you take the conductive vials and you load the acceptor solution into one vial. And then you can screw the union holding the polypropylene membrane onto this acceptor vial so that it fixes the membrane in place. That is what you see here in the picture. Then finally you tighten the vial firmly to fix it in place. And then you're ready to apply the solvent, which is 6 to 10 microliters. That is just pipetted directly onto the surface of the membrane. So you can see here, before we apply the solvent, it is white. And then after applying, it becomes transparent, which is easy to see when you get right. It only takes a few seconds, but if you wait up to 60 seconds, then you're sure it's spread out evenly. Then you load your sample solution into the sample vial, and you screw this to the other side of the union, and then you have one completed EME cell. You of course have the option of writing the sample name on the plastic bottom part here, or you can attach a sticker or whatever you use to identify your samples. You place the samples into the holder as shown before. The negative electrode, which is called the cathode, is facing towards the operator. So that means that if you want to extract a positive ion, then this should be your acceptor. You close the lid and tighten the screws as is seen here. And then you set the extraction parameters in the software. and start the extraction. And during this time, the white holder here is shaken to stimulate the extraction. While this is happening, the instrument will record the current that is going on inside of each unit. We will not talk too much about current today, but this is this you can use sort of like you would use HPLC pump pressure. It tells you something about the health state of the system, so that you can monitor for each position. And when the extraction is done, for example, after 15 minutes, then the instrument itself will shut off the voltage and agitation. You can disassemble the EME cells, and then you can just put a HPLC cap on this vial and put it into the autosampler. Or if you want, you can transfer the volume into another regular HPLC vial or 96 well plate, whatever you use in your system. So you can go quite quickly from extraction to injection. That really only takes a few minutes. Then we will go into the performance you can expect to see. And of course, performance can be many things. So the first thing that comes to my mind is selectivity and efficiency. And here we have illustrated this by extracting 90 pharmaceutical substances that cover a quite wide range of hydrophobicity, which we express by the log P. So you can see if we plot the extraction recovery, which is the percentage of the sample ions that were put into the sample that actually were transferred into the acceptor solution. If we plot that against the hydrophobicity and extract with one particular solvent called 2-nitrophenyl octyl ether, or NPOE for short, then you can see that you get sort of a window where things within this window are extracted with near 100% efficiency. So this we call the extraction window of this system. When you go towards more hydrophilic substances with a lower log P, these are not extracted, and this is because they are simply too hydrophilic to go into this hydrophobic supported liquid membrane, so they are discriminated. And on the other side, you can see that when substances get to a certain hydrophobicity, then they also are not extracted, and that is because they go very well into the hydrophobic solvent, but they just do not come out again. The interesting thing about this technique is that this extraction window we can modify and play around with. So if we say that this is the extraction window for this solvent using 50 volts of extraction potential, then if we decrease the voltage to just 2, then you can see that the window decreases in width. We can also make the window wider by using a higher voltage, at least that works until a certain point. So let's say you want to go to a higher log P for your analyte, then you can switch to another solvent that can handle higher log P values. Or if you want to extract something that is towards the more hydrophilic side, then you can take another solvent. And if you even want to go further down, then we can use some specialized system that relies on something called ionic carriers, which I unfortunately do not have time to go into in this webinar. So, of course, you can see that we can get high recoveries, but people often ask, is it repeatable, or is the precision good? And for this, we have taken a system where we have designed this to extract acidic substances, and this is from human plasma samples. So this is 31 acidic substances, 1 point for each, and then we have plotted the relative standard deviation against the recovery. And what you can see here is that as long as you do not get down into the very low recoveries, then you get quite good relative standard deviation of, in this case, it's around 5% for most of the compounds, above 40%. Another question people sometimes ask is how large of a concentration span this can handle. This we recently tested using the compound called verapamil, which is a pharmaceutical drug. And here we developed an HPLC method to monitor this, and then we extracted this in different concentrations. And what you can see here is at 0.1 nanogram per milliliter, which was about the lowest we could measure with the LCMS method, we have about 100% recovery, and this continues all the way up to 1 milligram per milliliter. It's only when we go to 10 milligram per milliliter that we see a saturation. So this is 10 to the 8th fold difference, which is much, much wider than most analytical detectors. So almost no matter what application you have, then this can work for that concentration range. Validation performance is, of course, also very important. Here I have taken one example, which is a paper published in 2021, where 12 psychoactive substances have been extracted from clinical plasma samples. And this is then validated according to FDA validation guidelines. I will not go through the entire table, but what you can just notice is that you get very nice linearity ranges, and the intraday and intraday precisions are also very nice, mostly less than 5%, very low bias or good accuracy, and you also have very low or no matrix effects even without internal standard correction. Cleanup efficiency, so how well do we actually clean up the sample matrix, is of course very important. After all, this is why we do sample preparation. And if we start from the biggest things you may have in your matrix, then we can consider particles and cells. And these are discriminated very efficiently just from the size of the pores in the membrane, which are about 0.2 micrometers. So any particles larger than that will by default not be extracted. And then the solvent itself will also contribute. So particles are not extracted at all. The second largest thing you can consider might be proteins. This we recently evaluated by using the same solvent I showed you before, NPOE, and extracted human plasma samples. And then we ran an HDS SDS page gel, which some of you may be familiar with. So you take your protein and apply it on the top, and then you also do some electrophoresis to transport the proteins, and then smaller proteins will migrate further. Then you have a ladder out here that shows you how big the proteins are. And if we look at the line D3, then you can see the unextracted plasma sample, of course, is completely overloading gel because plasma contains a lot of protein, about 80 milligrams per milliliter, actually. And then we have the EME extract, so the acceptor solution, which you can see is completely free of even a trace of protein. The last three lines here, D7 to 9, is then a comparison made for different protein precipitation methods. This was actually a little bit surprising to me, especially D9, which is protein precipitation with citronitrile, which I think for many people is their go-to method. And as you can see here, you actually have a lot of protein left after this. So if you have a few hundred samples of this injected, I can imagine why people's column do not last very long. But fortunately, we do not have that problem with EME. Another thing that can be troublesome when you run LCMS from blood-based samples are the phospholipids that are very well known to cause ion suppression. And these we have monitored by using an in-source fragmentation method where we just monitor this phosphatidylcholine backbone. So in the electrospray source, we can cleave off this bond, and then we get rid of all the variants of the R chains. So we just can monitor the 60 to 70% of all phospholipids that follow this structure. Chromatograms you see here are then from LC-MS, where the top one is a protein precipitated sample that of course do not remove any phospholipids. So here you have huge signals. Then if we zoom about 100 times on the intensity axis, we have different curves. The red one is for electromembrane extraction extracts, which are completely free of phospholipids. We have one peak here, but that peak is also present in the blank. And then interestingly, we have compared this to commercially available phospholipid removal plates. And we can see here in the blue-green trace that those actually had some traces left. So EME is also superior to that. Another thing you may wonder are then the small molecule endogenous metabolites. And here we have taken the same system as before and extracted a urine sample and run Hillick UV chromatography, which gives you a very rich chromatogram when you have unextracted urine. Then the green trace here represents the acceptor solution after we have performed EME with this system using 2-nitrophenyl octyl ether, where we also just have a few traces. Of course, if you modify your system to be more permeable to more hydrophilic substances, then you will extract more than is the case here, which is a system that's mainly suited for hydrophobic substances. When you then evaluate the matrix effects you get, I showed you in the validation table before that these are in general quite good. Then we have taken different methods, some that are suitable for basic substances, some that are suitable for acidic substances. And then we have taken a lot of different substances and extracted these and then checked the matrix effects. And in this case, 100% indicates that there is no difference between the matrix effect in the extracted sample as a neat standard. And you can see here that for all of the methods, they generally center around 100%, even without internal standard correction. And this is because that we get a very good cleanup, so you have next to no matrix effects in the electrospray ionization. The final performance parameter you can consider is then the green chemistry aspect, which many people perhaps do not think too much about today, but as we move along, then I think this will become more and more important and perhaps also regulated. This is an example that compares a liquid-liquid extraction, classical liquid extraction for amphetamines in urine to electromembrane extraction. You can see here that we went in with the same volume, and then we have to have some formic acid to adjust the pH, and we have two different solvents that are mixed together with EME. In total, this consumes 11 microliters of solvent. We have no use of inorganic materials or chemicals, and we can use just 2 steps to perform the extraction. And here we have already decreased the amount of organic solvent almost 100 times compared to classical LLE, and we need far fewer steps, which can make the procedure a bit easier to handle. Then we move on to how you can get started with electromembrane extraction, because this is something that is new to most people, and if you're an experienced researcher, then you will often have a good idea how to, for example, start with an SPE or LLE method. But to start with EME, the first thing you have to consider is the charge of your substance and the hydrophobicity. And both of these can often be obtained computationally. We like to use a web page called Chemicalize, which can calculate these things, and we find this to be very accurate to real life values. So we highly recommend this. Once you have considered these, then you have to develop a method which consists of finding a suitable SLM solvent and the volume of this, what sample and acceptor pHs you should have. These are general chemistry parameters. And then you have to find what acceptor volume, sample volume, agitation rate, extraction voltage, and extraction time to use. These parameters will be discussed in a upcoming webinar because there is a lot to say on each, which we don't have time for now. But you can read more in this documentation, which is available on the ETN homepage, or you can find a tutorial that we have published in an open access journal a few years ago that also goes through some of the same things. We have, however, also developed a number of generic methods, which are intended to allow you to skip the method development if you can find a generic method that matches the hydrophobicity and charge of your substance. The idea here is that you find out, is my substance positive or negative, and what is the log P, and then you will see that it fits within one of these quadrants. And then we have different generic methods that targets different quadrants. To help you make the determination, we have made a homepage called howtoeme.com. And here you're able to search a substance by its name or other identifiers such as a CAS number. For example, here I have searched methotrexate. And then this homepage will tell you, first of all, if EME is suitable, and second of all, it will suggest you the closest generic method we think might be suitable, and then you can start from that. There is also on the ETN homepage documentation that describes the specific generic methods and what kind of performance you may expect. As we near the end, then I have a few application examples, but I think in upcoming webinars we also will spend more time discussing dedicated examples. The first one is a proof of concept study where we extracted nicotine from e-cigarette juice. The purpose here was that these e-cigarette juices are regulated in the sense that they are not allowed to contain nicotine. But when people then analyze them, they find that many contain it still. So then, of course, it needs to be monitored. And in this case, we did a proof of concept study for some that were interested in measuring nicotine with LC-UV. They struggle with an overlapping matrix compound, which you can see here that overlapped nicotine. So we tried to extract nicotine from this e-cigarette juice by a sort of best guess EME method. So this was not very optimized. And you can see down here the chromatograms before and after the extraction. And you can see that we got rid of the matrix peak and we had about 100% recovery, which was quite satisfactory. The second example I have is then a little bit more towards the bioanalysis field again. And this is extraction of pharmaceuticals from tissue samples. And this shows you the classical procedure for analyzing tissue. You will take the tissue and then you will freeze it before you can do a simple pulverization. Then you would add a buffer to this and then do some homogenization to break up the tissue and cells. And then you can get on to do the classical extraction by adding a water immiscible organic solvent and shaking this, collecting the solvent before you have to do evaporation, reconstitution, and filtration to avoid the particles, and then you're able to analyze by LC-MS. But in the example I have here, they were able to skip the homogenization step and just took the simple pulverized tissue and added a buffer, put this into the sample and extracted this in one step before analyzing the acceptor with LC-MS. Specifically, they determined the anti-cancer agent called danirubicin and its metabolite danorubicinol in rabbit tissue. You can see here that these substances have an amine group here and here, so they can be positively ionized. To do the extraction, they took 25 milligrams of tissue and added 200 microliters of dilute formic acid and some methanol. That was a sample. Then they used another solvent I have not mentioned, but this is called DEHPI. Used 0.5 molars of acetic acid as the acceptor, extracted for 15 minutes at 15 volts for 25 minutes, and took the acceptor solution after this and analyzed by a fairly fast UHPLC MS method. And then they compared EME to the performance of their reference LLE methods. The conclusion of this comparison was, of course, that EME required fewer steps, which is nice, but they also saw no additional benefit of homogenizing the tissue before EME, so they got the same result if they did homogenization or not. And compared to LLE again, they saw improved recovery, sensitivity, and sample cleanup with EME. We can see here for both myocardium tissue and liver tissue that EME was superior to LLE for both metabolites, but in particular for liver tissue, EME was a lot better for this danirubicin substance. This might have to do with the fat content of liver, because EME is also able not to extract the fat. Finally, I have a horrible slide with a lot of other examples of published applications. There is a number of forensic applications, and these are nice if you want to see validated methods. We also have worked on extracting peptides previously. This is a little bit more premature. We have examples of endogenous metabolite extraction or pharmaceutical metabolite extraction. The vast majority of these examples are published with open access, so most of you, or not all of you, will be able to access these. I will mention that not all of these were performed in the ETN format, since some of these examples predate that, but the chemistry is the same. I also have a few examples that are unpublished of what we have used it for. We have done a few proof of concept studies, for example, where we have extracted alkyl phthalates and alkyl phosphates from liver extracts, or we have worked on removing phenol from a peptide solution. So this is an example where you don't extract the sample and then you are interested in the acceptor solution, but where you have something in the sample that you're interested in removing from the sample, and then use the sample for something else. We also have worked on extracting quad pesticides, which are permanently positively charged, so they are very suitable, and have shown that that can be done from juice. And otherwise, I can mention that we have some ongoing projects about PFAS extraction, bisphenol extraction, EME applied in what is called volumetric absorptive micro extraction samplers, which is should we say, the future of blood sampling. And then we have some projects that are more fundamental, like predicting the efficiency quantitatively. But we will come back to more applications in future webinars. So finally, I will like just to summarize and hope I have given you the impression that EME can offer a green, efficient, and highly selective sample preparation of, among other things, but not limited to, biological samples. And this is especially useful for drugs and their metabolites, but peptides, endogenous metabolites, metals and heavy metals, and preparative extraction applications are also possible. If you want to learn more, then I can direct you to the ETN homepage, etneme.com, or you can try this howtoeme.com webpage to predict if your substance is possible to extract. We also have another webinar coming up on April 8th. And then we will have so-called EME days, which are one-day courses. One will be in Germany on June 3rd, and one will be in Norway on June 5th. And these courses cover the principle in more details than I have done now. theory and fundamentals, and importantly, it gives you a comprehensive introduction to method development, applications, troubleshooting, the latest research, and also some practical training. So, as we have reached the end, I'll invite you to submit your questions to the Q&A session, and then we can take it from there.
Yes, thank you so much, Frederick. Can you see the Q&A already? We
Yeah,
have one
I see
question
the Q&A.
in.
Yeah. I'm not sure if everybody can see it, but I can just name the question. So the question is how long time it takes to optimize parameters during method development. And of course, that depends on what kind of substance you have and what your requirements for the performance is. But once you have found the solvent that works, which maybe you have to screen 5 or 6, but we are gradually moving towards narrowing that range of solvents by characterizing the ones we have. Then you might spend a few days developing the methods. Mainly you have to optimize the extraction time and extraction voltage. And extraction voltage can be because the instrument can apply different voltages to different channels, then you can actually do that in one run. So yeah, I would say a few days, but it depends.
Do we have any more questions there? Perfect.
Yeah,
Question also, if we have played around with the membrane's porosity.
That has compared to everything else, that has been explored fairly little. One limitation here is that it depends on what kind of porosities we have been able to get commercially. But we have played around a little bit with it, but there is not much difference if you, let's say, increase to 1 micrometer instead of 0.2. But it's something we are working on in parallel with the rest of our research.
Yeah, and I can also add that that's one of our main focus points for the next couple of years is to investigate and research that also. All right, I see it's 1 more minute. Do we have any more questions? Yes, let's see, we got one. You post. Yeah, it's a question about the 96 well support. Our goal is to launch the 96 well plates at the Analytica conference next year, so it's June 2026. But the good thing with this instrument that we have now, the 12 position instrument, it's that is perfect for method development, and we want to make the 96 well plates in a way that you can transfer the methods over from the 12 position system and up to and into the 96 well plates.
I can mention that we in our research group we have a sort of home built 96 well formats, and a lot of the not a lot, but some of the especially forensic applications examples that were in the slide are also performed in this format. So we know that works very well. And I saw there was one question about the slides, and of course we can distribute those, so you have all the examples.
Yeah, so send the slides and the presentation. We have a couple of more minutes for those that want to stay. Otherwise, just feel free to contact me directly, and we will answer all kind of questions you have for this kind of instrument and the technology itself. So yeah, before you all leave, I just wanted to thank Frederick and all of you guys for participating.